Welcome and good morning, dear students. Say yes. So this is probably the last day of our training program. This 11th of November. So we are very fortunate to have our principal investigator of this most prestigious NAHEP CAST project being implemented at IARI. We are also part of that particular project. So I would like to add my own words of welcome to Dr. Viswanathan Chinnaswamy sir, who is here to deliver a lecture on plant phenomics. And just to introduce him, sir did his graduation from TNAU and then followed by this post-graduation MSc and PhD from IARI New Delhi. And he had also gone to USA for completing the Boys Cast Fellowship at Professor Jiang Kang Ju's lab at Department of Plant Sciences, University of Arizona. Also, he is visiting researcher at this particular lab, that is University of California. Also visiting scholar at Department of Molecular Biology, Harvard Medical School, Harvard University. And he has got, so he is fellow of so many prestigious academies and societies, but just to mention a few, he is fellow of National Academy of Agricultural Sciences and Indian Society for Plant Physiology, Indian Society for, this NAS Associate Jawaharlal Nehru Award by ICAR for Best Postgraduate Agricultural Research. Many more medals are with him. Currently he is now occupied with Head Plant Physiology at IARI, and also he has got so many external funded projects and participated in international collaborations also. Many significant contributions he made in the field of plant physiology and in general plant biology. Sir has got several publications in Nature, very rare cases, in India. So he is our guiding force, for the entire, I can say, Indian agriculture in general, and in particularly in the field of plant phenomics. He has created with all his efforts, day before yesterday you visited the phenomics lab, and that's all due to his vision and contributions. And also he has got 3 patents. So we are really, sir, very fortunate to have you here today morning. So with this brief introduction, once again I request you to please deliver the lecture. Thank you.
Wherein the manual effort is reduced, the phenotyping based on the sensors, and where when you record manually make any observation, there is a possibility of variation because of our perception. So if we want to record the leaf color, everybody will record some different gradient. If you want to record the leaf rolling or leaf drying, senescence in the field, so everybody will record their own version. So there will be no commonality, and the quantitativeness is very less because human eyes we can see and detect variation, quantitative variation very easily. When you ask somebody to quantify, then it is very difficult. So maybe you may be seeing me, and after some time you ask somebody asks to display, you may say it's maybe about 5.5 feet height or something. Then if you say how much is weight or how much is this volume, what is the color, and if you ask someone to distinguish all the participants in the lecture hall, it will be very difficult. So in all these cases, the sensors play a very important role, where the phenotypic data acquisition is very precise, and the data is archived for future reference. So you have the data all the time for future reference. So why the science of phenomics came in? What is its necessity? For this, I want to give some two slide. So now, as you all must be dealing with DNA sequences, so getting genomic information is nowadays very easy. You might have heard that Genome project they are starting, wherein they are going to sequence 1.5 million species to the world. And also in India, there is a program on human genome, wherein you can register yourself with IARI, and then your genome can be sequenced for free of cost. So genomics is now it is very easy thing to do. So you do sequence and you get the sequence information. So how to use the sequence information? That is the major question. The ultimate purpose of doing genomics is to use the DNA sequence information and the sequence variation for improvement of crops, animal or human diseases, and many more things. So in case of plant science, the scientists they met in 2012-13 they all the years academics they met, and they thought that what should be the vision for the plant science research in the coming decade. So they set a decadal vision. So in which they proposed that we have the ability to create from plant genomes in diverse environments. So this is one of the ambitious goal or mission. So you have the sequence information. From sequence information, you have to predict how the plant phenotype will be there, and more importantly, diverse environment. It is not that in one environment how it will be there. It's how the plant is going to behave in different environments. Or other specific trait, for example, grain quality will be there if you grow it in Delhi or indoor or outside. So all these things, if you have to predict, you have to initially establish the genotype-phenotype relationship, and then you have to see how this genotype-phenotype relationship changes in a different kind of environment. If you remember, phenotype is the interaction of genotype and environment, which you will see in some of these slides. So once you predict this, then you assemble the trait. So at present we do a lot of phenotyping, 3-year, 4-year phenotyping in the field, and we try to establish some phenotype relationship, and then try to use that information for conventional marker assisted field. So still we are far from predicting the ability of the plant, apart from the few functionally validated genes. So guys, you know about 3,000 genes are functionally validated. So for those genes, you know the function, like if there are two for some one or two alleles for which the characterization has been done. So rest a lot of things to do, which needs a lot of phenotypic data, like huge amount of phenotypic data, and then association studies between phenotype and genotype, and then later you use this data to predict the phenotype. So once you know the phenotype, then assembly of different traits to solve the problem. Like in every area and every person may have different requirements. To solve that requirement, you have to, in different way, you have to assemble the trait. Then you environment. So basically, like this is the force, the sum of the atoms, molecule, macromolecule. So many phenotypes are possible for a given plant, given environment. So unlike the genomics where you can sequence and get the genome information, phenotype information is difficult to collect for all the phenotypes that a plant can produce in the given environment. Phenotyping, when we say phenotyping, it is the measurement of a set of phenotypes. So it is on the genome-wide phenotyping. We select some traits and then we phenotype out. So phenotyping is, when we say phenotyping, it is nothing new, as all of you have done phenotyping. It is the first maybe the scientific activity human beings started. So from the domestication, then later crop improvement. Initially, mainly relied on the phenotypic selection when the genetics was not known. So it was phenotypic selection, and then later came in once the Mendelian genetics was known and the DNA structure and function was elucidated, then it is genetics-based selection and recombination breeding has been started. Then later, for functional genomics also it is very important. That is, if you want to, if you know that, right, that is if a plant is short or a plant produces good quality of fruit, then if you want to know what is the genomic region that contribute to that, then you have to do map-based cloning, then you write the region, and then completely validate. So in all those cases, you will be doing phenotyping. Then assembly of desired trait, that's what we do in the breeding, either it is cross breeding or any other approach where you have to assemble different phenotype, wherein you need precision phenotyping. In case of molecular breeding, where you use marker assisted selection, in that also significant amount of phenotype is involved, because some of the traits which are linked to the trait of your interest may also come in your recombinant inbred population or the backcross population. So in that you have to see what is the phenotype of your interest and select the near isogenic lines. Besides this, so for optimization of cultivation practices, also you need phenotyping. If you decide, say, row to row spacing and plant to plant spacing in a field, you have to optimize what is the optimal leaf area index required to harness the solar radiation. So you have to do a precise phenotyping, and then you decide what should be the optimum combination, rows to row space or plant to plant space, how different plant dimension should be there. So all these things can be optimized, or how much amount of nitrogen has to be applied if you want to have